So in chapter 4, we're going to be looking at eukaryotic cells and other microorganisms, and they're comparing their cell structures. The first eukaryotic cells appeared about 2 to 3 billion years ago. They were very simple, but then they evolved into complex organisms. Only certain eukaryotes are going to be traditionally studied by microbiologists. We're going to look at protozoa, microscopic algae, fungi, and then some helminths. Helminths are going to be worms. And those aren't technically, by the way, microscopic, but they do have microscopic portions of their life cycle. Almost all microbial eukaryotic cells will have a cell membrane. They're going to have a nucleus. They're going to have your endoplasmic reticulum, your Golgi complex, vacuoles, cytoskeleton, cytoplasm, and glycocalyx. And you're going to want to be comparing these and be able to compare the eukaryotic cells, which is what we see here on the left. And I don't think this picture is in your text. I think I found this somewhere else. And compare them with a prokaryotic cell. That's going to be real important. You're probably wondering, why do we care what the cell looks like? Well, I've told you several times already, if you are trying to break into a bank or a fortress, you've got to know the layout. You've got to find the weak spots and how we can break in or destroy the cell or stop it from reproducing. If we don't know it front to back, how it's laid out, then we can't fight it, right? Okay, so you can also see we've got mitochondria here and lysosomes, where the prokaryotic cell, you can see we've got a capsule. We've got ribosomes as well. They're a little different DNA, but it's in nucleoid regions, not in a defined nucleus. Plasma membrane, cytoplasm. So some types of eukaryotic cells do have chloroplasts, but not all. Some have cell walls, usually kind of the same. Like a plant is going to have a chloroplast and a cell wall, but a human or animal cell does not. Some have flagella. The only flagellated cell in the human body is sperm. And then there are, of course, some other appendages that can be used for movement, like cilia. Let's talk about some of those different appendages. We have cilia and flagella, okay? And they're a little different than bacterial flagella, all right? They both function for motility. And remember, motility is movement, and it allows microorganisms to find nutrients and move towards substances. We have positive stimuli like sunlight that you would want to move towards, or food sources, or maybe some harmful substances that you would want to move away from. Protozoa, algae, fungi, and animal cells have cilia or flagella. Can have anyway. Eukaryotic flagella, like I said, are very different from bacteria flagella. The eukaryotic flagellum is going to be much thicker. It's more complex, and it is covered by a cell membrane, and it is also covered with microtubules in this what we call a 9x2 array, which sounds very complicated, but you've got these two little sections, are singlet microtubules in here, and here you have doubles, and then there's 9 of those, okay? That's what the 9x2 array means. So during movement, the flagella are going to kind of whip back and forth. Cilia are similar to flagella, but they're shorter, and there's a whole lot more of them. They will usually surround the whole cell, okay? Cilia, then they're going to beat back and forth almost like strokes or oars, you know, like paddles, okay? And so they're going to assist with movement. Like there's a ciliated protozoa that's going to move and use that movement to help it feed, or sometimes even with filtering of certain substances. Next is the glycocalyx, and that can be found on many different eukaryotic cells. This is going to be that outermost layer. Sometimes it's called the extracellular matrix, and it is going to be composed of different polysaccharides. Sometimes it can be a slime layer. Sometimes it can be a capsule, similar to bacteria, and sometimes it just appears as a network of fibers, and so it can kind of take these different forms. The glycocalyx functions in protection, adherence to surfaces. That's an important one. So it helps to protect, but it really helps it to stick to things a lot. And it also helps to receive signals from other cells or signals from the environment. So as we said, some have cell walls. Helminths remember, are worms, and protozoa do not have cell walls. Animals do not have cell walls. Fungi have cells that are going to be rigid. They provide support and shape, but they have a different composition than bacterial cell walls, so they're put together a little differently. They have an inner layer of a polysaccharide, and remember, poly means many, saccharide is sugar, so lots of sugars in there. And they can have chitin, which helps it to be a little more rigid, cellulose, and then sometimes some glycans that are part of the outer layer. So the cell membrane is usually going to be this bilayer that we see over here. Okay, this is a phospholipid bilayer. All right. It's going to have these proteins embedded in it and throughout it. It's going to have what we call sterols here, which are cholesterols that help to give the membrane more rigidity. And that's really important in cells that don't have a cell wall. They've got to have some way of having a little bit firmer structure, okay? And so these are what we call selectively permeable membranes. In other words, they regulate what goes in and what comes out of the cell, which is very important. The nucleus is probably the most prominent and one of the most important organelles of a eukaryotic cell. It's separated from the cell cytoplasm by an external boundary that's called the nuclear envelope. It's composed of two parallel membranes separated by kind of a narrow space there. It is perforated with very small pores that are kind of regularly spaced. And this is going to be formed at sites where the membranes unite, and things are allowed to go in and out of those pores, like RNA. Macromolecules, they can go through the pore to the cytoplasm and vice versa. The nucleolus is basically the nucleus inside the nucleus, and so a lot of times people get those mixed up, so be sure that you differentiate and understand the difference between those. It's inside the nucleoplasm, and so nucleoplasm is basically just another word for cytoplasm, but the cytoplasm that's in the nucleus. Its chemical composition is just a little bit different than the regular cytoplasm. And this is where ribosomal RNA is made. It's also where we put together ribosomal or collect ribosomal subunits. Now, this diagram over here is important because I find a lot of times, and I remember as a student, not really understanding the difference between chromatin and chromosomes, okay? So what is the difference? Well, it's the same thing, all right? But structure determines function, and so when things change, you're going to see that we will give it a new name. Like if we have an enzyme, and you won't understand, maybe it depends on what you've had. If you've had anatomy, you know what I'm talking about. An enzyme and a substrate, when they're bound together, we have to give it a new name and call it the enzyme substrate complex, okay? And so it's the same material, it's just in a different format, okay? So here we have our chromosome. That is what we think of as a chromosome, and it looks like a big X. But what we have to remember is this is very, very tightly coiled up, and this is a very poor representation of that. But so this is kind of how we have that X, is it's all coiled up. And so this is showing you here how this is uncoiling, okay? And so when it is uncoiled, or when it's coiled up, we call it chromosomes, okay? But chromatin is going to be the DNA that is kind of unwound, and it's going to be wrapped around a histone protein. So it's wrapped around basically these yellow blocks here, this histone protein. And that's what's used to help it to coil and uncoil. And what I think of when I think about histone proteins is like Christmas time. And you have the Christmas tree lights, and you have the little green or black plastic that the lights are wrapped on, and you wrap them on there, you use them, and you unwrap them, and you put them on the tree, and then you wrap them back on. So it's really just something to coil around, okay? It's just something to coil around. So that's the only difference. Next, we have the endoplasmic reticulum, or the ER. We have two types of that. We've got the smooth ER and the rough ER. The smooth ER is over here, And so basically the difference is one, have a little bit different function, and two, some have ribosomes and some don't. And so the smooth ER do not have ribosomes, okay? The rough ER does have ribosomes. And so the function for the rough ER, ribosomes are help to do with protein synthesis, and so the function of those is usually going to deal with proteins, okay? So they're going to transport things from the nucleus to the cytoplasm, okay, to head out towards, to leave the cell. Often it's going to be proteins. The smooth ER is a little bit different. It doesn't have the ribosomes, and it functions more in making things that aren't proteins, like lipids. So making lipids, okay? And the way to keep that straight is to think about those ribosomes. Ribosomes are where we make proteins. And so the rough ER has ribosomes, so protein synthesis, and the smooth ER does not. So it tends to make lipids, which are fats. Next, we have the Golgi apparatus, and this is kind of like the post office. And so once it gets the proteins, it's going to modify them a little bit, pack them up, and then ship them off to where else they need to go. And so it's got these pita-like flattened disc here, and these sacs, this disc-like sacs are called cisternae, and these are always, it says, closely associated with the ER. That just means they're going to be nearby, okay? So transitional vesicles here from the endoplasmic reticulum are picked up at the face of the Golgi apparatus. Proteins are going to be modified in the cisternae. They're going to have polysaccharides stuck on them. They're going to have lipids stuck up on them. And then they're going to be put into these little vesicles, which are actually, you can see here looking at this one, they actually pinch off from the membrane of the Golgi apparatus, and then they're sent on their way, okay? So they're put in these little lysosomes, basically, which can be transported out of the cell. So that's why I say it's like a post office is going to pack it up and ship it off. All right, so here is an example here of the transport process. So the nucleus is here, all right? You have your endoplasmic reticulums. You're going to go through your endoplasmic reticulum. The Golgi apparatus is then going to take something, it's going to modify it and change it and pack it up. So let's say there's a segment of genetic code of DNA in the nucleus down here. It's going to be copied into RNA because the DNA is too large to go through the pores, and we don't want to risk it leaving. We want to keep it safe. So it's going to go through the pores, and it's going to go to the endoplasmic reticulum. And then there are going to be specific proteins on the rough ER that are deposited into the lumen. They're transported to the Golgi apparatus next. Proteins in the Golgi, are they going to be modified? Okay, you might stick a sugar on them. It depends on what their function is. You're going to change them just a little bit, and then they're going to be packaged up into vesicles, which can then leave via exocytosis. Exo meaning leaving, cyto meaning cell. So endocytosis is going in, exocytosis is going out. We have different types of vesicles. We have lysosomes, and we have vacuoles, and we also have peroxisomes. Excuse me. A lysosome is a vesicle that's going to bud off the Golgi apparatus and continue doing things. It's going to have a variety of enzymes involved. It can digest food particles. It can protect against invading microorganisms. It can also help to clean up and remove cell debris. So lysosomes are often going to help clean things up. So you can think Lysol, lysosome. We also have the peroxisome, which isn't on here, but it's a type of lysosome. It doesn't come from the Golgi apparatus, so it's a little bit different. And that actually helps with detoxification of substances like a reactive oxygen species. And it takes it and forms it into H2O2, which is hydrogen peroxide, okay? The vacuole is going to often contain a food vacuole you can have, and it can digest things. It can merge with a lysosome and do some work there. So it can digest solid particles, it can excrete them or store them, okay? You find these vacuoles in a cell that does phagocytosis. Phago meaning eat and cyto meaning cell. So basically digesting or eating a cell. And so something will be taken in, and it's going to, the vacuole, the enzymes in there are going to help to break it down or digest it. This illustration here shows a food particle that is going to be engulfed by the cell. The contents are going to be in the food vacuole. They're going to be digested. So this is phagocytosis. You're going to have a merger with the lysosome and the vacuole, and then we call that the phagolysosome. So remember, I said if you change something, it gets a new name. Okay, so you're going to have merger of those, and then it's going to be digested with those enzymes. So this is just kind of showing you that process. Mitochondria, you'll find, are going to be very important. Now, you're often told, students tell me, I ask them, what's the function of mitochondria? And they say, well, it's the powerhouse of the cell. Okay, well, what does that mean? Can you tell me what that means? Because if you don't know what it means, that's useless, okay? So people say it's the powerhouse of the cell because it generates energy, okay? So this is or it helps to generate energy. It makes ATP. This is composed of a smooth, continuous kind of outer membrane with a folded inner membrane. That folded inner membrane is the cristae. And on the cristae is where cellular respiration, the last part of cellular respiration occurs. It's got the enzymes and all the electron carriers, and we'll talk a whole lot more about cellular respiration. It's got everything that's needed to complete that process embedded in the membrane. So we're going to extract chemical energy that we find in food and nutrient molecules and store it as ATP, which can be used as an energy currency for our bodies. Okay, this is a very unique organelle. It's got a very interesting history. It can divide independently of the cell. It has its own mitochondrial DNA, and so that's a lot of times you may hear that you get the majority. Now, we used to teach all of your mitochondrial DNA from your mother, but we found out recently that you get some from your father, but they have their own DNA, which is kind of crazy. They have circular or what we call plasmid DNA, and some of them have bacteria-sized ribosomes. And so we have 70S and 80S. So the ribosome size is a little bit different in a bacterial cell versus a eukaryotic cell. Well, these are eukaryotic cells. And so we'll get into more a little bit later about the endosymbiotic theory, which is really kind of fascinating theory. We'll spend just a very short period of time on it, but we can't spend much. But you might look up some stuff on that. It's really kind of interesting. All right, so chloroplasts, these are going to be found in algae and plant cells. These are also capable of energy conversion, except what they're going to do is basically the opposite. They're going to take energy from the sun, which is going to be turned into chemical energy using the process of photosynthesis. Okay, we're going to have oxygen gas as a byproduct, which is very helpful for us because we need that to breathe. These do resemble mitochondria. But they're larger. They have, of course, special pigments, which makes them green. They're a little bit more varied in shape. All right, I already said it, but eukaryotic ribosomes, remember, are larger than bacterial. The eukaryotic ribosome is 80. Bacteria have 60S, and some even have 40S subunits, and that just refers to size. And the function is going to be protein synthesis. Okay, that's very important. You can find these attached to the endoplasmic reticulum, but they are also found kind of freely floating out in the cytoplasm. They do appear inside mitochondria. They're also found inside chloroplasts. They can actually be found in these short chains in what we call a polyribosome. So this is a ribosomal subunit here. This is going to be where proteins are made, and this is the polypeptide or protein coming out here. And so you can actually find these in these short chains all together. The cytoskeleton is composed of 3 different types of filaments. We have our actin filaments, our intermediate filaments, and our microtubules. They function to anchor organelles, help move things like RNA, proteins, vesicles. They allow shape changes to occur. The actin filaments are going to be longer, thin protein-type strands. The intermediate filaments are going to be more rope-like structures, and the microtubules, of course, are going to be little tubes, or long, hollow tubes, I should say. And these are pretty much made on an as-needed basis, and then when they're not being used, they're broken down. And so I'm probably going to make a little video talking you through the secret life of the cell and showing you how some of these portions of the cell function together, okay? And that's a really, really well done video, and it's going to really help you to see and kind of wrap your mind around how all of this stuff works. So this is just another look here about how these things are kind of in the cell. You've got the actin filaments here, and then the intermediate ones, and then your microtubules are more like tubes. Kind of makes sense. This is a really good comparison chart of the structure and function of organelles in each of the different types of organisms. Now, you would really serve yourself much better to make this kind of a chart yourself. I would put bacterial, archaeal cells, and then eukaryotic cells, and then go down through and list everything and look it up for yourself. If you actually look it up for yourself and make this yourself in the textbook, then you will have done the work. You will know the material. And if you just trust me and try this, if you just try to rote memorize this, it's harder and it's boring. But if you go through and you do the work yourself, put this together yourself, and then you can come back to this and check it, okay, and see what you got right or wrong, and if you left something off, but you definitely need to know all of this information. So here is this endosymbiotic theory, and this is where scientists believe that millions of years ago, a bacterial cell actually parasitized another, basically a descendant of a eukaryotic cell, and became a permanent part of the mitochondria. And so there's several different types of evidences for this. We're not going to look at it in great detail, but some of this is because they have their own DNA. Okay, that's one of the reasons why they think this has occurred. Okay, so it's really kind of a cool theory.